Hello there and welcome back. This is our third and final video in our video series for Chapter 4 on genetic inheritance. My name is Miss Delia, and in this video, we're going to go into and finish 4.3 genetic mapping. If you're ready, let's go. These are the learning outcomes for this topic, so I hope that by the end of this lesson, you should be able to define what's genetic mapping, and you should be able to calculate the genetic distance or the map units, and last but not least, identify the position or order of genes along a chromosome based on the data that you are provided with. Okay, when we talk about genetic mapping, we're actually relating to one of the contents in the previous subtopic, which is on linked genes. So linked genes are two or more genes that are located on the same chromosome. So like in this example, we have the green labeled one, which is A, and the pink labeled one, which is B. When we talk about genetic mapping, we are putting them on the chromosome. Kita memetakan gene-gene yang kita tahu berada pada kromosom yang sama. So you need to look at how far are they, like these two genes, and where exactly are they located. Before we go into the whole how to calculate, we need to first give some definitions. Okay? So sebelum kita masuk kita punya isi kandungan, kita cakap dulu pasal definisi. Genetic mapping is when we take the proportion of recombinant offsprings resulting from a dihybrid test cross and use that to calculate crossover value. So kalau dihybrid test cross itu ada tidak ikut Mendel punya hukum, dia tidak ikut 1:1:1:1:1, then kita boleh ambil data itu untuk kira crossover value ataupun COV. COV is a measure of linkage, and if linkage occurs, the distance between the genes can be determined. COV adalah untuk sukat linkage. Maksudnya, kalau ada COV, means kita tahu kromosom itu, dua gene itu berada pada kromosom yang sama. Dan kalau dia berada pada kromosom yang sama, kita gunakan data daripada COV untuk tahu jarak di antara dua gene tersebut. The COV is a mean to look at the map, locate relative positions of genes on the same chromosome. So kita gunakan dia untuk memetakan dan mengenal pasti posisi gene pada kromosom yang sama. And genetic mapping can only be carried out if the genes are linked and if crossing over takes place. Gene yang kita tengok itu mesti berada pada kromosom yang sama. So that is what is meant by linked. And if crossing over takes place. Kalau crossing over tidak berlaku, kita tidak akan dapat data untuk membuat calculation ini. If the linked genes are situated very close to each other, it cannot be separated by crossing over during meiosis. Kalau dua gene yang kita tengok itu sangat rapat, masa crossing over pun dia akan terikut dengan kawan dia juga. Dia susah mau terpisah. Kalau if the two genes you're looking at is very close, it's hard to separate them during meiosis. And if they're situated further apart, kalau jarak antara dua gene yang kita tengok tu jauh sedikit, lebih banyak kemungkinan untuk mereka terpisah semasa crossing over. The possibility of crossing over to occur is higher. So this is something that you need to remember. And therefore, the crossing over frequency is directly proportional to the distance between two genes. Semakin dekat dua gene itu, semakin susah crossing over berlaku untuk memisahkan mereka. Semakin jauh dua gene tersebut, semakin mudah crossing over berlaku untuk memisahkan dua gene ini. That is what is meant by these sentences. Okay. So what we need to do next is I need to teach you about the genetic mapping formula. Untuk kita kira jarak antara gene, kita gunakan formula ini. Crossover value is equal to total number of recombinant offspring divide by total number of all offspring times 100 or 100%. 1% of COV is equivalent to 1 map unit. Okay, bila kamu sukat-sukat-sukat, hasil kiraan kamu 1% bersamaan dengan 1 map unit. Map unit adalah unit yang kita guna untuk sukat jarak antara gene. So you can use map unit, which is m.u. Or you can also use centimorgan, cm. Cm dia Tengok betul-betul di bawah sana tuh, cm. C kecil, M besar. Unit ini dinamakan sempena saintis yang mengkaji link gene. Okay, so you can use map unit, you can use centimorgan. Okay, all talking is no use. Let us try with an example. So, ini ada satu contoh. Kamu boleh cuba-cuba jawab sambil kita tengok video. Let's try to calculate genetic distance. Below are the offsprings from a test cross done on Drosophila with gray body and normal wings. So, gray body and normal wing kita cross dengan black body vestigial wing. Sebab ini adalah test cross. Hasil daripada test cross, gray body normal wing 965, black body vestigial wing 944, gray body vestigial wing 206, black body normal wing 185. Kemudian, soalan minta kamu di atas sini, soalan minta calculate the distance between the gene control for color body and wing size. Ayatnya tunggang terbalik, tapi basically apa yang dia minta, sukat jarak di antara gene yang mengawal warna badan dan size sayap. So, let's assume the allele for body color is big B or small b, and allele for wing shape is big V or small v. So, soalan bagi juga kamu, okay, mari kita gunakan B dengan V untuk body size and wing shape. So, kita gunakan formula yang kita dapat tadi, okay, ini formula kamu perlu hafal lah. Hafal formula ini, crossing over value is equal to total number of recombinant offspring over total number of offspring times by 100. What is the total number of your recombinant offspring? Please look at the data we have here. So, kamu boleh tengok, total number of recombinant offspring is supposed to be the smaller numbers. Bilangan yang lebih kecil itu. So, here it is 206 and 185. Masa kamu buat kiraan, letaklah di bawah macam tu. 206 plus 185 divide by everything else. 965, 944, 206, 185. Times by 100. Kira-kira tekan calculator, kamu akan dapat 391 over 2,300 times by 100. And you will get 17%. Apa maksud 17% ni? Lepas kamu kira-kira, kamu dapat 17%. That means, jarak antara gene yang control body color, which is B, and the distance between that and the gene that controls wing size, which is V, is 17 map units. So, 1% adalah 1 map unit. 17% adalah 17 map unit. Kamu juga boleh gunakan centimorgan. Okay, so that is how you calculate using this formula. There is another type of question that you will see for this subtopic. Ada dua bentuk soalan. Bentuk soalan pertama adalah macam ini, dia suruh kamu kira. Bentuk soalan kedua adalah dia bagi kamu data macam ini dan dia minta kamu lukis peta kromosom. Okay, the other type of question is that they're going to give you data like this, a lot of numbers, a lot of letters, and then they're going to ask you to arrange these genes on the chromosome and basically draw a map of the chromosome. When you get this kind of data, the tip is to start with the highest COV. Ini adalah yang persen-persen ni, adalah orang itu sudah kira menggunakan formula dan dia dapat nilai ini. So all of these are COV values. Okay, so we're going to start with the highest COV. Now you can follow with me on this one. Let's look at these. Yang paling tinggi COV value di sebelah sini adalah 40%. So the 40% is between gene R and gene Q. So I'm going to draw a line. When I want to draw a map of the chromosome, I draw a line. And then I put the R and the Q. R dengan Q sebab dia punya COV paling tinggi. Kemudian saya akan lukis ini. To write 40, kamu boleh tulis 40%, 40 MU, 40 cm pun boleh. And then I'm going to do the second one. The second one is P and Q. P and Q 35%. So, tidak sepanjang 40. P dengan Q 35. So, saya letak macam ni. 35. Third largest COV is 30%. 30% antara R dengan S. So, it should be around here. R dengan S. And then I'm going to use the remaining smaller numbers to check my answer. So, yang tinggal adalah 10% antara Q dengan S dan 5% antara P dengan R. Saya cuba letak sini. Okay, nampak macam betul dan nampak macam betul. So, when I check and see that my map makes sense with this data, I am done with this exercise. Kalau kamu cek, kamu punya map, kamu punya peta kromosom selaras dengan maklumat yang diberi dalam soalan, means siap sudahlah jawapan kamu untuk soalan jenis macam ni. So you should draw a lot of these kind of maps. Okay, so it's a very short subtopic and that is pretty much it. That's all for genetic mapping. We relate it to linked genes. Kita kaitkan dengan linked gene dan kita belajar untuk kira crossover value, and then we learn to draw a genetic map. Itu sahajalah. Okay, what you should do next is now you should complete your worksheet for 4.3 with your genetic mapping worksheet, which you can find in the Google Classroom. And we also have your tutorial exercise for chapter 4, which you can find in the website. I'll also provide a link if you're unable to find it. Yeah, please try to complete all of them if you can, and we will discuss them on our next lesson. Yeah, so that is pretty much it. Thank you very much for your time and attention. With the end of this video, we have finished chapter 4. And yeah, please do practice for this chapter. It doesn't make sense if you don't do any exercise at all. And I'll see you again soon. Thank you and goodbye.